Okay, make sure you check your email. I just sent you an email this morning with some a list of the tests you need to do for your unknown report and some instructions, and then also a query to you about the exam next week. So take a look at that. The results are the lab exercises. There are some things you need to look at 24 hours. I'm going to give you a few minutes into the period. Remind me again, phenylalanine it says to look at, but we're going to leave that until Friday, so don't bother looking at phenylalanine right now. I think arginine was one, is that correct, Kendall?
Yeah.
And then lysine was another
Lysine,
one? Ornithine. Okay, so look at those three. Lysine I haven't had a chance to tell you yet, but we need to do a follow-up test if you get a positive lysine, and we'll do that tomorrow. So just look at it for any color changes in arginine, lysine, and ornithine, and then put that back in the incubator. Urease, if you get a positive result, you're done. But if you don't get a positive result, you want to put that back in the incubator. Okay? So remind me about 20 till if I haven't stopped. Give you some time. Okay? So last time we ended up talking about some of the nasty diseases that staph can produce, and one of the take-home messages is that staph is a pretty nasty pathogen because it produces multiple virulence factors. That is also the case with streptococcus, and there are several species of streptococcus. One of the slides later on will show you some of the more common species. Probably the species you might have heard of most is Streptococcus pneumoniae, and that causes bacterial pneumonia. Some of you maybe have heard of Streptococcus mutans. Austin, have you heard of that? You hope so? That's your seminar, right?
Yeah.
Okay. Kettle caries. Okay, toothcase. We're going to be talking a little bit about some of the diseases caused by a particularly nasty form of strep called strep pyogenes. There are all kinds of species of streptococcus, and they're a little bit hard to identify. And several years ago, probably several decades ago actually, a researcher by the name of Rebecca Lansfield, started differentiating streptococcus species on the basis of some antibody testing. And she put streptococcus species into various groups that are now referred to as Lansfield groups. And they were given letter designations. They were designated as groups A through H and K through B. Not sure why we missed the middle there. But typically, Lansfield groups A and B have some of the worst pathogens in them. Streptococcus pyogenes is a Lansfield A streptococcus. One of the ways that you can differentiate Lansfield groups A and B from the other Lansfield groups is if you plate those bacteria out on agar plates, and then you put bacitracin discs on the growth, the Lansfield A and B will be inhibited by bacitracin, so you'll get that zone of clearance. The other Lansfield groups don't do that. So that's a fairly easy way to differentiate between those particular groups. Strep pyogenes can cause all kinds of unpleasant conditions. Streptococcus pyogenes can cause pharyngitis. What should you think of anytime you hear the word itis? Inflammation. Okay, inflammation of the pharyngeal region. Okay, a red sore throat. But Streptococcus pyogenes is not the only agent that causes pharyngitis. There are a lot of viral infections that cause pharyngitis as well. When you start getting the streptococcal infections, particularly strep pyogenes, oftentimes it's more than just the inflammation in the red raw throat. Usually you're also looking for these pus-filled pockets on the tonsils. Streptococcus, like the staphylococcus, can produce leukocidins and other toxins that kill white blood cells. And these pus pockets are basically accumulation of dead strep and dead white blood cells. So if you see something like that, you probably suspect strep throat, bacterial cause as opposed to viral cause. But those rapid strep tests that are available now will give you that particular diagnosis very accurately. If you don't treat Streptococcus pyogens, it can then manifest itself into a systemic infection where the toxins that it produces starts affecting capillaries in the skin and can lead to this whole body rash. Sometimes the skin just starts to slough off as the rash develops. You may know what this particular disease is called. Right up here. This is called scarlet fever. Okay? And this is a consequence of untreated strep infections. So it's not a thing to mess around with. You know, I mean, you really should figure out if you have symptoms of a strep infection and find out what it is. If it's viral, then obviously you don't treat with antibiotics, but if it isn't, you're going to want to get some antibiotics on board. Another possible consequence of an untreated strep infection is rheumatic fever. Have you ever heard of rheumatic fever before? Rheumatic fever results when heart valve tissue is damaged as a consequence of the infection. What's interesting, though, is this is not a result of any toxins that the bacteria produce. This is an immunopathogenesis. This is friendly fire. This is your immune system actually doing the damage. The reason that this happens is that there is a phenomena going on called cross-reactivity. And what happens when you get infected with strep is it elicits an immune response. And strep antigens elicit the formation of antibodies, which is good. That helps to eliminate the strep infection. But there are some antigens on heart valve tissues which are very similar to those strep antigens. So the antibodies can then start attacking the heart valve tissues and cause heart problems. Okay? So you don't want to mess around with strep infections and not treat them when you really do have one. This is another very nasty disease. This is called necrotizing fasciitis. And the infection causes severe inflammation of the fascia, which is that kind of connective layer that helps to hold skin to lower muscles and things like that. And so the skin really starts to slough off and peel off. This looks like kind of like a bad tattoo. It's really all of that skin that's being sloughed off. You may know what the common name for this particular condition is called. This is the infamous flesh-eating bacteria that you might have heard about in some of the popular news. So again, pretty nasty disease. Well, this is a summary of some of the common strep species that cause various infections. And if you look up here, Lancefield group A, Streptococcus pyogen, has a pretty extensive list that can cause several very serious diseases. Okay? Okay, so we're talking about why microorganisms are able to cause infection. It's because of the virulence factors that they possess. What typically is going on during the infectious process? What's happening during the course of a typical infection? Well, if you look at the number of microorganisms or intensity of symptoms over time, a typical infection will run a course that looks much like a bacterial growth curve. And typically we divide this particular infectious cycle into various stages. The earliest stage is the incubation period, and that's where you're kind of cooking up the population. But at that point in time, you're asymptomatic. You're not suffering any symptoms. That's typically because there aren't enough bacteria there to produce enough of those virulence factors to really affect your tissues that much. Then you get into this stage called the prodromial or prodromal stage. And that's where you go, man, something's not right. Okay? I'm feeling lousy. I'm off. And that quickly transitions into the illness phase, and that's where you get the most severe signs and symptoms. And that corresponds with rapid growth of the microorganisms, production of toxins, other virulence factors. Hopefully, the condition will be acute. Acute diseases are self-limiting. They run their course. And eventually we start getting into a decline. And the decline in symptoms is associated with the decline in the number of microorganisms. And eventually we get into a convalescent state where you're recovering. Sometimes the convalescence is fairly prolonged, especially if a lot of damage has been done. So even though the pathogen has been resolved, you can still have a long period of recovery. And sometimes, as I mentioned earlier, you may never completely recover. You might have long-lasting consequences like paralysis. That is referred to as being a sequelae. Well, this general course of events occurs in virtually all infections, but the width of these particular bars varies significantly from one type of pathogen to another. And one of the things that is of a lot of interest is this incubation period. Okay? Because during the incubation period, even though you're not showing symptoms, you are potentially a source of infection for others. So it's very kind of timely to know what are the incubation periods of some common diseases. Okay? Some are pretty short. Influenza has a very short incubation period. Some are very long. Look at leprosy, a 10 to 30-year incubation period. Would that suggest that this disease is very communicable? You know what communicable means? Can you pass from one person to another? Okay. No, it's not. And yet, look what we did to people for a long period of time when leprosy was a feared disease, because we were afraid that it was highly contagious or very communicable. It's not. There are various types of infections that can occur. A local infection occurs when the infection occurs primarily at the portal of entry. So acne would be a good example of a nice localized infection. Sometimes pathogens will spread from one infected site in the body to another, but they'll remain localized in that new site. So sometimes, for example, you might get a tooth abscess, and then that infection spreads to the heart, for example. That's called a focal infection. And sometimes the infection agents get into either the blood or the cerebrospinal fluid and give you a systemic or a whole body infection. So that scarlet fever we saw, where the rash was entirely over the body, would be a good example of a systemic infection. Measles is another systemic infection. Infections are also characterized as being primary or secondary. Primary is caused by the infectious agent itself. Secondary infections are a consequence of a primary infection. So, for example, urethritis, inflammation of the urethra, bladder infection. What might you do? Take antibiotics. What might that do? Suppress your normal flora. Actually, it was interesting, sidebar, Kendall ran across an article that he sent me. There's a study that was done. Remember when that was done? It was Switzerland. That said they did some comparisons, and their conclusion was that taking even one course of antibiotics could affect your normal gut flora for up to a year or more. So that's really some strong evidence that we should be very careful about prescribing antibiotics, and you shouldn't have the attitude, well, I'm going to get an antibiotic just in case it might be a bacterial infection. because we're finding out more and more of the consequences of destroying and disrupting that normal gut flora. But in the case of bladder infections, it's not uncommon for a secondary infection of yeast to develop, because now we're wiping out with the antibiotics those antagonistic bacteria that are part of your normal flora that typically keep the yeast in check. Another thing that can happen is that you can have chicken pox, for example, and have lesions. And if you scratch it, you might have some staph on your fingernails, for example, and then that can give you a staph infection. If you scratch open that lesion, that would be a secondary infection as well. Pneumonia is another common secondary infection following a viral influenza, for example. So again, here's some terminology. I'm not going to go through all of this, but these are some infection speak that you should be comfortable with, in terms that you should really know. Some of these we've talked about already. Some of them we will be talking about in just a minute. Diseases typically are associated with particular symptoms. And that's convenient for treatment. Many of you have had experience with Dr. Posek, if you ever did the Johnson Memorial rotation. And Dr. Posek says almost every time I hear him talk to students, one of the things I really love the most is diagnosis. To look at a set of symptoms and signs and to be able to then deduce what the infectious agent might be or what the underlying cause is. So what's the difference between those two, symptoms and signs? Symptoms are sensed by the patient. They tend to be more qualitative. They tend to be more generalized. Signs, however, can be detected or measured by an observer, including a physician. And they tend to be more quantitative in nature. I mean, somebody can say, I'm nauseated. But if a doctor sees you throw up, then that is a sign as opposed to a symptom. Okay? Someone can say, I feel hot. Okay? But if they take a thermometer and see if your temperature is 103, then that would be a sign. Palpating your lymph nodes to see if they're swollen. Okay? The term syndrome refers to particular signs and symptoms that are associated with particular diseases. And so that's really what a good diagnostician is looking for, is those particular pieces of information that give you a sense of what's going on. One of the symptoms that is most commonly associated with any disease or any infection is inflammation. Well, at some point in time, the pathogen is probably going to have to leave the body. It entered through a particular portal of entrance, and so it's probably going to leave by a portal of exit. Oftentimes the portal of exit is different from the portal of entrance. And as you can see here, there are a variety of ways in which a pathogen can move from one host to another. Individuals who have the pathogen and are carrying it around can be the cause of disease to another individual. We typically characterize people who are harboring pathogens and have the ability to spread them into several categories. And these refer to situations where somebody is a carrier of a disease. Carriers may show symptoms, but oftentimes they don't. Oftentimes the carrier might be completely asymptomatic. Their immune system is strong enough and robust enough to keep the disease tamped down, but they're still potentially shedding pathogens. And if an immunocompromised individual is exposed to those pathogens, they might cause a problem. Incubation carriers. These are people who are walking around that are totally asymptomatic, they'll eventually will develop symptoms, but they're just brewing the disease right now. Some people will still be able to transmit a disease even when they're in the convalescent state. So their symptoms are pretty much dissipated now. Chronic carriers harbor the pathogen continually in their body, even after they've recovered and while they're totally asymptomatic. I mentioned typhoid Mary. She harbored the typhoid bacillus bacteria in her gallbladder. Okay? And those pathogens got into her digestive tract from the gallbladder and were shed into feces, and she was a food handler. So she was able to transmit that disease as a chronic carrier through that fecal transmission route. In hospitals, we have to be particularly concerned about passive carriers. And those are individuals that aren't harboring the disease. They don't fall into this particular category of care. But they can unintentionally transmit an infectious agent from one patient to another, oftentimes mechanically. There are some diseases that are not indigenous to humans, but can be spread to humans. Those particular diseases are called zoonosis, or zoonotic diseases. Typically in these, the animal is where the pathogen typically resides. But they can be transmitted to humans, although they typically won't move them from humans to other humans, or from humans to animals. So humans are kind of a dead-end host as far as the pathogen is concerned. There are various helminthic diseases like tapeworms that are zoonotic. There are various protozoan diseases, including malaria, fungal diseases. Anybody ever had ringworm before? It's a zoonotic disease. But there are also bacterial and viral diseases that are zoonotic. Probably the one that you've heard of most is rabies. Okay? Rabies primarily resides in an animal reservoir. That's the place where you find the pathogen in its normal state. But humans can be infected by bites. We used to have a faculty member here, Dr. Poley. I don't know if any of you have ever heard of him. He's been gone a while. But he was a parasitologist, and he used to teach a winter term class that concentrated on transmission of diseases from pets to their owners. And these were also known diseases. So, I mean, if you took this class, you would never let a dog look you in the face. It's horrible diseases that can be transmitted that way. I don't know if that's considered. Probably technically it is, because H1N1, is in mainly birds. Yeah. But it can be transmitted to humans, yeah. You did? That's not good. Okay. Have fun training. Oh, will you be able to come back and get your look at your stuff?
Yeah.
Okay. When we consider a disease communicable, it's sometimes convenient to break communicable diseases down into methods by which they're transmitted. Are all diseases communicable? Can you think of an example of a non-communicable disease? You just can't get it from another person? Cancer? Yeah. Thinking bacterial in nature. That's correct. Tetanus is a non-communicable disease. Okay. Communicable diseases are typically broken down into methods of transmission from one individual to another and categorized as either direct transmission or indirect transmission. Direct transmission includes both horizontal and vertical transmission. What does horizontal refer to? me to you, okay, person to person. Vertical transmission refers to mother to child, okay, transmitted across placenta. The most common routes of direct transmission are contact, where the mucous membranes of one individual come in contact with the other. Kissing, mononucleosis is sometimes referred to as the kissing disease. Droplets are a major way of transmitting communicable agents from one individual to another. If you want to see something gross, here's a high-speed picture of a sneeze. And look at all of those droplets that are being put out into the air. And if you basically suck up some of those droplets, that's considered to be communicable or a direct transmission, even though the mucous membranes aren't in direct contact. Now, we're going to see in a second, for indirect transmission, is this guy could have sneezed into an empty room, not on an individual. Okay, if somebody sneezes in your face, that's pretty much the direct transmission. But if this guy sneezes in an empty room and then leaves, these droplets can become suspended in the air in the form of aerosols. And you could walk into that room later on and breathe in that aerosolized particles, and that would be considered indirect transmission. I'm not sure why, but biological vectors are typically referred to as direct contact. So like a mosquito bite, the transferred West Nile virus would consider to be direct transmission. I've never seen the logic in that. Indirect transmission typically involves things like food or water, the oral fecal route. So food handlers, for example, if they are harboring pathogens, can transmit to the food. Hepatitis outbreaks oftentimes originate in restaurants. And if there has been an outbreak of hepatitis, typically epidemiologists will try to trace that back to a source. And they'll try to find out who the individuals are so that they can then be isolated. I refer to the droplet nuclei here. Animals can sometimes shed aerosol particles from their feces into the air, and if you breathe it, you can get infected. There was a disease called hantavirus several years ago, which was primarily in the desert Southwest, and they traced it to feces from a type of rodent that was living in that desert environment. One of the common sources of indirect transmission are fomites. Fomites are objects that come in contact with individual skin and become contaminated by being handled. Doorknobs are a good fomite. You know what probably the number one fomites are right now? Phones, cell phones? No. But anything that is a kind of an inanimate object that gets touched by one individual and can pick up bacteria is considered to be a fomite. I mentioned in an earlier context nosocomial infections, more commonly now referred to as HAIs, hospital-acquired infections. And these are of great concern because many people that are in the hospital are already sick. And so if they get contaminated, particularly with resistant forms of pathogens, it's a bad mixture. There's kind of an intersection here in this Venn diagram, if you like, of events that lead to nosocomial infection. And one is the presence of microorganisms in a hospital environment. Yeah, they try to be as clean as they can, but I mean, hospitals are a magnet for pathogens. Sick people come there, okay? Another part of the triad are immunocompromised patients. A lot of sick people with weak immune systems. But also transmission of pathogens between staff and patients and among patients is a big part of it. Something as simple as hand washing can have a very significant effect on the rate of nosocomial infections. And hospitals actually now are required to report the incidence of these particular type of infections. And hospitals have become pretty strict about hand washing. You'll see hand sanitizer, waterless soap in all of the rooms. Doctors are constantly encouraged to, rub stuff on their hands as they're moving around the hospital. The nosocomial infections really plummeted when antibiotics were first developed, but as organisms started to develop resistance, people kind of lost the good practices that prevented infection from the first place, because basically what they would do is they would give patients kind of a prophylactic dose of antibiotics on the way out the door, so treat you in case you picked anything up. But now prevention is really the big part of the strategy now, and typically there are hospital committees, which are infection committees, and their role is to monitor infection rates, to monitor compliance, and to suggest strategies that will limit these particular type of infections. Here you can see that a lot of those nosocomial infections are urinary tract infections. Catheters can be a big source potentially of nosocomial infections. You probably don't need, give me 2 more minutes. I think 15 minutes is plenty. Okay. Surgical wounds, skin, and this slide gives you, whoops, I had a slide here that showed you the organisms that typically were associated with nosocomial infections. Okay, that's going to lead us into the last topic of this particular section. Maybe this is a good place to break. Epidemiology. So we'll talk a little bit about what epidemiology does, how this information is reported, why it's useful, and why this is such a backbone of the U.S. public health system. So that's where we'll pick up on that. Okay, so don't worry about phenylalanine. You really need to look at lysine, ornithine, and arginine, and then just stick all of those back in again for tomorrow. So take a picture of your 24-hour. With the lysine and ornithine, it's very helpful to put those next to the controls so you can kind of see what positive and negative results look like. Because some of the color changes, particularly in the lysine, are a little subtle.